thanks a lot for being here. Thanks a lot for presenting me. And I'm very glad to be here. Today, I would like to talk about the fact why it is now a very good moment to invest in biotechnology. And I hope I can give you 2 or 3 examples if the time is, if we have got enough time. I think they are very interesting. And as I mentioned at the beginning, we are there close to a breakthrough. But first of all, as I mentioned before, since 20 years I am writing about this branch, and there's one sentence that appears in a repetitively manner, and in the newspaper column I often mention that. That's the sentence. It's very interesting what's happening in the biotechnology, but I'm afraid that I don't understand that. And yes, I understand that it is very difficult to understand. And it is easier to send some students on a bicycle as a shop helper. I know these are limits, but the last 20 years I noticed that they have really, there's a lot of investors are afraid of biotechnology. And I think now is a very good time to change this. And this, of course, has something to do with the corona pandemic. Who of you knows what a spike protein is? I know now everybody here in this room, and most of you also know, well, roughly at least know, what it is an R number. And first time I was at the supermarket and somebody talking at the phone, talking about about DNA, I was really surprised. In the NDR, we have got a podcast about the DNA and the podcast of Dr. Kekulé. So it is really coming into public. These are very good podcasts, and it's a lot of science. So I think step by step we are changing the idea of biotechnology. Biogen thought it would be a very good idea to represent which is the rare disease spinal muscular atrophy is, the SMA gene. So a couple of years ago, this would be more shocking, and now people seem to be more interested. I think now it is a really good moment for that. So this has something to do that now we are less afraid of this topic. Oh, I've lost. So I'm working for those editions, Euro, Börse Online, Courage. These are the newspapers. I'm once in a while collaborating, especially the one on the left side. I'm not giving you an advice about an individual investment. I'm more or less giving you examples. Why is it now a good moment to invest in biotechnology? This has something to do because we have got now more knowledge. Mr. Leber already mentioned that we are doing and publishing and researching more than ever. We can see this on that graph. These are the publications in the last years. Since a couple of years, it's more than 1 million per year. And we have got not only biomedical publications, we have some that are going into physics and science, natural science, and all the other publications that are not audited. So we have got really a lot of additions. Anyway, just having only the knowledge, that's not enough. We have to work with this knowledge, and we need a broad scope. This means from the doctorate that at my time, once in a week, pass through the library and copies the most important facts for the own research. And today it is very different. Today you can receive all the information as you require, and just with one click you have got all the data. This works worldwide. And on the other side, machines can do something meaningful with all these data. I have collected this graphic from a paper where the artificial intelligence is using the signal waves in a cancer disease, and at the end the AI identified different paths that weren't known before. We don't have this high knowledge. We have got also more options what we can do with this knowledge. And the thought behind is that we have got more options, of course, for possible medication, and knowing the system around, we are committing less failure. Maybe we can also have a look at how this ends in a success. So the probability to launch medication more efficiently at the market, well, we are still at the beginning, but we see a tendency. Bringing substance to the market, we have to do different clinical test phase, and the probability that this all occurs is really low. 5 to 10,000 molecular connections, well, one is really successful. So the probability of success from molecules is represented by this graph. From 10,010 on, the orange lines is phase 1. There's not happening a lot. The probability is under 10%. In phase 2, we see an increase, 11–15%. Could be better, but we are at the beginning. And then we have got the phase number 3, where we see a positive tendency with a success 50 to 60%. And this is something we will observe in the next years. And this curve should increase if we have got more knowledge available. So this costs also a lot of money. We have heard about that beforehand, and the good news is that the branch has really enough money. We never had that much money for the companies as we have right now, 28 billion, and on all levels it's increasing. We have venture capital, that's the blue graphic, the orange are stock market launch, and Then we have got the follow-on financing. If major capitalists will close their money flow, even though a company could survive a couple of years and could invest and research a couple of years. And nothing is worse for a company than the need to save money while researching. So, on the other hand, we need also the statistics of the American Food and Drug Administration. What are doing the administrations since 2005? In total, the number of medication that were accepted, and the last years would grow if we consider also the vaccinations. In the last years, we create new ways to amplify the companies, and we have got several specificas, and it's more a cooperative process. That means the administrations and the companies are focusing together and looking for new solutions. So having a forecast, this is Mr. Albert Bourla, Slim Tirici, and is maybe more famous, but this is the CEO of Pfizer, and he told us that what has happened now with COVID should be possible with other diseases as well. And of course, this has something to do with the quick research we were able to provide. And all this experience we collected in the last years should be also help us for the future. What is the idea behind? So why did we find so quickly a vaccination? Well, Pfizer and BioNTech worked highly risky. Even when it wasn't clear, they already built production capacity. That's something very exceptional. And I think it will not happen again. And second point is that the administrations were very open-minded and collaborated and accepted the data relatively quick. And this was not extremely an exception, because we are working in this way since a longer term. So,
well, and the third point, that is actually the most important one. We have done many things in parallel. Studies and experience that you do normally sequentially, we did in parallel. And without reducing safety, we did everything at the same time. And I think that is something that will also happen again in the future. All right, fair enough. To give you a summary, I believe that the industry is in a very good position, and for the next 10 to 15 years, we will bear the fruits of the bases that have now been established. So this is a good moment to get in. And now I should like to show you one aspect of research that I think is very interesting, and that is the targeted protein degradation. And what exactly is protein degradation? So how many of you have been at home a lot during the past couple of months? I suppose many of you, and you have probably noticed that you in fact do create lots of waste. You go shopping, then you prepare food and you buy food, and then you have to find out when are they open, when do we do meet and collect. So we order a lot of stuff online, I live right downtown Munich in a building with 10 to 12 apartments, and after a few days the bin is filled to the brim. And then it did occur that somebody came down and said, hey, too much paper. But I don't want to take my 5 Amazon packets back up again, so I just put it right next to it. And then this is a self-accelerating process. So it creates a bit of a cluttered picture. My nieces built this for me. So I didn't want to look at pictures for Playmobil, but then if you don't want to see them, get yourself a cup of coffee. But I think this is easier to understand than the reality. But in fact, it does look like that. People put their trash outside the bins, so there's a lot of fly tipping. And the waste trucks in Munich, the bin man will not pick up anything that is outside the trash bin. So the bin means this is what we can take along, and that has been paid for in terms of the waste disposal. So what does this have to do with biotechnology? Well, we also have our own type of waste disposal in every single cell of our body. And this is a very big and fascinating molecule. So old proteins or proteins that you don't need in your cell anymore can be reduced, and then that can be put to use properly. And again, our body doesn't do it in an unselected fashion. It does it in a targeted way. It needs a signal. And this signal is a very small molecule, and it's called ubiquitin. And there are enzymes that are inside the cell, and they choose the proteins where they say this can be taken away, and then they attach the right protein. And to trigger this mechanism, Tsikhanova, Dutschko and co. got the Nobel Prize for that. Since that mechanism has been known, the question is, couldn't that be done in a much more targeted fashion? And proteins that we don't want simply mark as waste. And I think of a whole bunch of illnesses where it would be great if we could test that. For example, Alzheimer, Parkinson, cancer. So there you have proteins in cells that cause problems. But also for cancer, of course, this can be extremely valuable, because the concept of cancer is there is a mutated cell giving the signal, break the cell. And if you could remove that signal for the division of cells, then you would be able to cure cancer. So people have been doing a lot of research on that. You have to imagine it in such a way that the first generation of medication, was, recognize which is the protein that you want to get rid of, and then there's another one, and then you have to form the gaze as a project, and then proteolysis carcitinera, if you want to remember that name. So it has the potential to be a new type of medication, because it does the degrading. As you see from this list, so it's not only for cancer, but also for other types of diseases. Second, most types of medication that we have today are based on the same principle. I use my active agent and either I activate or I inhibit, one of the two. So you have an active center of the molecule that is being bonded with or a receptor, and that's how it starts. But in many diseases, what happens is that these active centers or these receptors go through a mutation, and then my medication won't work anymore. And there are many proteins that are important here that don't have an active center. So you need some kind of structure in proteins, and then it works with marking the degradation, marking the trash. And if I bond with an active center or a receptor, you can imagine my drug has to stay there, because it goes out again, then of course it won't work. The process will be annulled and everything will stay the way it was before. And with PROTACs, it's not like that. So one can go to a proteid and mark it, and take the next one and mark it, and goes to the third one and mark it. And if it works like that, I can use very low doses. So it's a new mechanism. It can connect with molecules that so far have not been approachable. And there are a number of companies that are active in this field. Avinas and Chimera, these are names that you know from the United States. Avina is basically, they're the pioneers. That was founded from an ex-Yale professor who had started this development, and they're all there in phase 1 or phase 2. So in the next 12 to 24 months, we will see if it works the way it is being described here. If you look at this list, you see that these are all cancer diseases. So if I'm a good scientist, I start something completely new. I don't go into a complicated system like Alzheimer or Parkinson, where nothing has ever worked, but I go into a system that is well known and well researched, like prostate cancer or breast cancer. Because the advantage, if something doesn't work the way it's supposed to, it's easier to find out what it was that did not work or why it did not work. Exactly. So now we have the names of a number of big pharmaceutical companies, Bristol Myers Squibb, Sanofi. So you see companies are trying to get in there either by in-house research or they acquire other companies. And that's great for the smaller companies that can make money in those corporations. And the moment good data are available, we will see takeovers, which is, of course, always something that is good for the shareholder. So, in conclusion, I think if you look into the depth of a fund portfolio, you will also see one of these companies listed there. Maybe not in the top range, but it has the potential. And if companies like Avinas or Chimera, they have maybe $2–3 billion, that's not really cheap, but does not reflect the overall potential that is still in the making there. So these are the protein degraders. The next field I wanted to present to you is the gene therapy. For the past 2–3 years, it was the hot topic. Everybody's talked about corona. When everybody talked about corona, And KRT, KT, these are therapies. These are therapies against blood cancer, because these are therapies that are extremely successful with people who have this disease, who have been trying different things and it didn't work. And now we have success rate of 50, 60, 70, 80%. And this is for people who were basically close to death. And how exactly does it work? We heard a lot about the immune system, and so this is a way to fight germs and other causes of diseases. And that's exactly the problem with cancer, as you know. These are cells from the body, and the immune system does not normally want to attack its own cells. And these cancer cells are very smart, and they hide before the immune system. So even if they had had a special unit to fight cancer cells, and it's like you having a stadium full of football fans, and you know that there are a few terrorists in there. And it's just like in this picture, they look exactly like everybody else. There's no way you can easily detect them. But if you had a gadget, like a special X-ray glass or something like that, to see them, then that would be easy. You would say, there's one here, where's one there, where's there one on the other side. And that's the way it works with CAR-T. So you take some C cells, you test them. So CAR-T has the same principle. So, and then you have something that receptor that will connect with the cancerous cells. It works well, but there are also some major disadvantages. There is a whole bunch of side effects. In 40% of the cases, you have the so-called cytokine storm. So the immune system goes into overactivity. It's difficult to control. It can be controlled, but it is still a life-threatening situation. And then there is neurotoxicity. The making of a CAR-T therapy takes about 4 weeks, and it has to be individually manufactured from his own T cells for each patient. So if it is a very aggressive disease, it could be that in those 4 weeks he will die before you even start the therapy, or he's so sick already that his C cells are not so fit and you can't make a product out of that. And of course it is a lot of effort. 2 years ago I was in Fraunhofer Institute for this activity in Leipzig, and that's where the CAR-Ts for Novartis are made. And when I was there, I think that of the 320,000 euros that the therapy costs, anything is actually for Novartis. I mean, you are talking about 6, 8 clean rooms, and over weeks from 2 people, they only made 1 therapy. So there are 2 people fully protected, the way you know that from Hollywood movies. They work around the clock in a very, very difficult setting, and then they prepare the CAR-T therapy. That's why you want to change this therapy, modify it in such a way that it doesn't have to be individualized, but can be something that you take out of your fridge, basically. There are some steps forward, but it is very difficult. And now there's a new technology. Those are the natural killer cells. They're also part of our immune system. Well, there's the normal part that you're born with. They are very strong. And those are the natural killer cells. And then there's the inactive part that takes a bit longer before it actually gets going. But the advantages is that they work completely different than the CAR T cells. So you can use the killer cells from anybody. You don't even need a donator. You can take stem cell lines, anything. And then these NCAR cells don't have any side effects. So it's really a difference. For the CAR T therapy, you have to stay in hospital for 10 days to 2 weeks. And you have to have some doctor on call all the time because the storm may start. But this is something that is in an outpatient setting. The patient comes in, gets the cells inoculated, and then can go home. And the effect, well, you can put in these special receptors, but the CAR cells actually work without even that final modification. And the question is how long it will last and how good it is. And we see that now. Just now, the therapeutics in the US published a study, and there were patients that had gone through thousands of pre-treatments. So 11 had been treated. With 8, there was a remission. 6, there was a total remission. It's a question how long that will last, but this cell therapy is NK cell therapy is extremely promising, and I think they will be available for many, many people because they're not as difficult to manufacture as the CAR T treatment. Well, we have to see how long the effect will last. Many of these patients get a stem cell transplant anyhow, and this may be a therapy to cover the time, to bridge the time until you actually have a donor for the stem cells. And the NK cell therapy can be repeated time and again because it's available. And that, of course, makes this therapy quite interesting. And there's a couple of companies, Affimed from Heidelberg. They don't do their own NK cells, but they make molecules that help create the NK cells. You can add them, but you can also use the ones that are already in the body of the person. And again, I think it's a very promising technology, worth your while to look at. These are fairly small companies here. But the big companies are there too, as you see the names here. And if that is really true, what we have now in terms of data, it probably is one more tool in the toolbox. And you can make good money with it. So thank you very much.
Thanks a lot to you. Julia Gross, please come to us. Stay there at that desk, please. So we have got keep the distance. Yes, I'm already vaccinated, full vaccinated. So, Julia Gross. A lot of people watching us are, most of them, investors who maybe have not that much experience with biotechnology. What is, well, your approach to deal with biotechnology? Well, again, you should not put all your money in one basket. The smaller the share, the more risk you have, the smaller the title is. There are a lot of industries that are very successful. For instance, a phone company from Switzerland. If you want to be a little bit risky, you can read about it, and there's a lot of documentation that is easy to understand. And you should not think one German value will revolutionize the whole world. Maybe you should have a look to who is cooperating with whom, and that gives you a lot of about. Well, it's a quality figure who's collaborating with whom. So we have got very little companies here. You presented some of them, and which of them will become big? I have not counted them. But maybe most of the companies, of the small companies, are still there. Well, when you do your research, you talk to the people, and getting you so close to these companies, yes. I talk to them directly if it is possible, with the manager directly, and if not, well, there are different analysts and managers that I contact, and I ask them for further ideas. Just when I have a conversation with one of them, so just a smaller part will be used for an article, but to talk to experts, it's always good. You have got the feeling that right now, as Dr. Leber mentioned, there's happening a lot. We have more knowledge, the research is quicker, the results are easier to access to them. So yes, I think it's the same as Dr. Leber said. Well, we haven't agreed on that beforehand. It's just, well, a fact. So the cancer research, you think we will have a big breakthrough soon? If it's working on corona, why shouldn't it working on cancer therapy? Well, that's very optimistic. I think this will take some time. Cancer, since decades, is a very innovative area, because you can do very little small studies when people are very sick. What I have heard recently very often, the neuro and other diseases similar to Alzheimer, depressions and so on, this is a very important topic as well. So we are getting back to the nature. We are using different substances gained from the fungus against depression, and we think that's very helpful. So backing to the genes, to the substances we can gain from the nature. There are a lot of companies that are popping up that are using these kinds of drugs. At least they are drugs. They are doing their research on them. Maybe we can use certain components to create a new medication. And this was off limits for a long time because of the laws. And I think this started with the cannabis boom, that now we have got investors that are accepting this kind of research. And I'm very optimistic that some of these projects will be very successful. These are very natural substances that have a high potential. And again, Alzheimer is a very interesting topic. Biogen has already gained some has been accepted by the Food and Drug Administration. So I think we need more, we need 10 years of research to have a kind of breakthrough. due to the acceptance of Biogen, it will take some time to have some breakthrough now, because again, researchers are looking for the amyloid plaques and have a look at that. And even 2 years ago, we already knew that it isn't effective enough. These medication for there are projects that are not only looking at that specific protein. Again, a question. Can I ask you some questions, even if you're not working in science directly? Yes, I try always to answer all questions, yeah, as far as it is possible. And as long as this is not a question why I still have BioNTech in my investment portfolio. These are questions I cannot answer. Biotechnology and the big pharma companies. There was still a rumor that there are some limits, that the pharma industry is still interested to maintain the existing medication, and that the pharma industry don't want to support new developments, because, well, I think this is not happening right now. Maybe in individual cases, because you have got maybe a blockbuster that's very popular, and of course companies try to maintain the claim as long as possible and as the patent is running. So, but generally, pharmaceutical companies need these little companies for certain researches, for a certain expertise, and this is a really good business model to cooperate and step by step acquire these little companies. Which company is for you really good and doing a good job? Roche is very good. They have acquired BioNTech, and they have been very clever. It's a couple of years ago, by leaving this company alone, being independent, and maintaining the spirit of this little company. But right now, there are not a lot of differences. Where will be the added value? BioNTech, Pfizer, we can see that. Pfizer is going into the world and organizing all the administration and logistic level, and research and production is BioNTech. So we have got half of the economy on BioNTech. So the euro will have a higher value on the industry. Where should I invest? Of course, the little company, you have got a higher effect, especially the examples that I mentioned. These are very small companies, and if it works out what they're doing with these waste removal and the K cells, then we have got a very, very good added value. You cannot see that in Roche, but this is, well, this is investment that works together. So the former companies will need this flexibility from the smaller ones, and so we have got. So what's biotech and what's pharma? This is they are very close by.